Hello, my name is Dr. Roderick L. Roll, and today I will conclude our discussion on the history of microbiology. We will now talk about the modern days or the modern era. So here is our concept map. We have the early years, the golden years, and the modern years. During the modern age, three questions were addressed. How do we defend against disease was question one. What are the basic chemical reactions of life? Question two. And question three was, how do genes work? Here is our first question, which is broken up into three subtopics. How do we defend against disease? The first one we'll look at is chemotherapy. Chemotherapy brings us back to Paul Ehrlich. Ehrlich searched hundreds of compounds looking for one that would work against diseases. This search for the magic bullet created the field of chemotherapy, which is how chemicals can be used to treat disease. One chemical was finally discovered by Alexandra Fleming in 1928. This famous compound is called penicillin, and it is a type of mold. Much later, in the 1940s, Florey and Chain purified the chemical and used it to treat bacterial infection. So Fleming discovered it, but Florey and Chain purified it so that they can mass produce it. Another magic bullet was discovered by Domagk in the 1930s. He discovered a sulfur drug called Prontosil. Later in 1935, these two people discovered that Prontosil had a sulfanilamide component, which gave the compound antimicrobial properties. This led to the discovery of numerous sulfur-containing drugs. And I have the sulfanilamide compound in the picture below. Sulfanilamide was used to treat fungal and bacterial infections. Currently, it is mostly used to treat urinary tract infections because the compound will accumulate in the urine. Since the compound accumulates in the urine, when you urinate, if you have a urinary tract infection, now you will bathe the urinary tract with this drug because it accumulated in the bladder. The next component to our question of how do we defend against disease is serology. Behring and Kitasato both discovered the existence of chemicals and cells within blood that fight infections. We're going to extensively talk about this later, where these individual cells, like the basophils and neutrophils, actually fight against foreign invaders. So their discovery gave two answers to how we defend against disease. Serology is the study of blood serum. Some notable components in serum are antibodies and white blood cells. The antibodies are those Y-shaped structures to the left, and some of the white blood cells are over there. Some are mast cells, neutrophils, macrophages, and so on. C. How these chemicals in the blood defend against specific pathogens are referred to as immunology. So when we study how those chemicals actually destroy the pathogen, we are studying immunology. The second question gave us the field of biochemistry, but the concept started with Pasteur and Buchner's experiment on fermentation. Biochemistry looks at the chemical processes within living organisms. Some such chemical processes is the metabolism of a sugar called glucose. You learned a little bit of this in general biology when you took glucose and broke it down into pyruvate. There's 10 actual enzymes that help this to occur. So you therefore learned biochemistry when you learned how this process occurs. Then once the pyruvate was created, pyruvate was shuttled into the mitochondria and further enzymatic reactions occurred. That is still biochemistry. The third question is how genes work. This question led to the evolution of 4 new fields of study: microbial genetics, molecular biology, recombinant DNA technology, and gene therapy. The first one we'll look at is microbial genetics. Microbial genetics is the study of inheritable information in microorganisms. When we learn about how the organism on the left has a plasmid that protects it from a drug, and how that plasmid can be replicated, transferred through this conjugation tube into a F- cell that does not have the plasmid, now it will give this protective status to the F- cell. So this is the study of inheritable information in these microorganisms. One of the first studies that occurred on this was with Avery, McLeod, and McCarty. They performed an experiment on Streptococcus pneumoniae and determined that genes were contained in molecules of DNA. Two other scientists, Beadle and Tatum, established that genes' activity is related to the protein function. These two scientists performed the research on a bread mold called Neurospora. The second question, or the second field, was molecular biology. Molecular biology combines biochemistry, cell biology, and genetics to explain cell function on a molecular level. Linnaeus Pauling used gene sequences to determine taxonomy. So molecular biology is the major that I had in graduate school. So as you can see, molecular biology can be used in a taxonomic approach to help place organisms in their respective categories, to see similarities and differences between organisms. Mr. Wolsey used RNA sequencing technology to propose 3 domains. These domains are archaea, bacteria, and eukarya. The third field that was created from this question of how do genes work is recombinant DNA technology. Recombinant DNA technology or genetic engineering is when geneticists manipulate genes in microbes, plants, and animals for practical applications. We can take, for example, in this image, DNA from a human. So in this picture it says foreign DNA. We can excise that gene by using restriction enzyme EcoR1 and EcoR1 here. You can use restriction enzymes. You can cut that fragment out. You can use that same restriction enzyme on this plasmid to open it to create these sticky ends. Insert the DNA into the vector. Allow it to reanneal. And now take that modified plasmid and shuttle it into a E. coli or bacteria. And now the bacteria can mass produce whatever gene that was isolated from the human. So this is genetic manipulation or genetic engineering. We'll now discuss 10 genetically modified foods that were modified using that same technique I just discussed. In the early 2000s, when I first started looking at genetic manipulation of the bacteria Bacillus thuringiensis, only a few farmland crops used this technology. But now, as you can see, how it has catapulted into the mainstream farming technology. More farms and more farms are using genetically modified seeds to plant these resistant strands of whatever seed it is. The USDA approved genetically modified crops. You see, the list is not that large, but you see that corn makes up a lot, and soybean and cotton. I will now discuss a few of them. The first one is corn. Genetically modified corn turns up in many different products in the US. And corn on the cob is the least of it. This crop is used to produce many different ingredients such as processed foods and drinks, including high fructose corn syrup and corn starches. But the bulk of the genetically modified corn grown around the world is used to feed livestock. So when the cows go from the farm to the feedlot, right before they go to your dinner plate, they're going to be beefed up. They're going to be fed a high diet of corn. This corn is genetically modified. The next one we'll look at is soybeans. The second largest U.S. crop after corn is genetically modified soy. It is used primarily in animal feed and in soybean oil, which is widely used for processing food and in restaurant chains. In fact, soybean oil accounts for 61% of Americans' vegetable oil consumption. So if you use vegetable oil to cook foods or to eat salads, you are consuming genetically modified product. Cotton. Much of the genetically modified cotton is turned into cottonseed oil, which is used for frying in restaurants. So again, we are using genetically modified products. Potatoes. Very recently approved genetically modified crop is resistant to bruising. So they genetically modified it so that it will not bruise and may produce less of a cancer-causing chemical called acrylamide, even when exposed to high heat. Papaya So read through that one. Squash. So I just wanted to mention a few of them. Canola, genetically modified canola is used to make oil for cooking as well as margarine. So some of your canola oil and margarine is genetically modified. Alfalfa. In a controversial decision in 2011, the FDA approved the commercial use of genetically modified alfalfa that contained a gene making it resistant to herbicide. This crop is used mainly as hay for cattle. Another newly approved crop is this apple. Sugar beets. More than half the granulated sugar in the United States come from genetically modified beets. In some of my research in the early 2000s, we used genetically modified corn. We spliced genes from a bacteria called Bacillus thuringiensis and put it into corn. And the corn is able to now resist insects such as grasshoppers. This is the process. The Bt gene is cut out and put into the corn, and then when the worm or the grasshopper eats the foliage, the leaves of the corn, it immediately dies. The last topic or the last category is gene therapy. Gene therapy evolved while we try to answer this question of how do genes work. Gene therapy occurs when scientists insert a missing gene or repair a defective one in an organism such as a human. This video explains it. There's a really cool example of how they use this gene therapy to help cure this young lady of a debilitating disease called sickle cell anemia.